All right, so now we're going to talk about the genetics of common diseases with multifactorial disorders. And so just first, this is a table with common disorders with the approximate prevalence and then how they are treated. So you could see cleft lip and palate has about 1 in every 1,000 births, and you can treat that with surgical repair. Clubfoot is again going to be the same, and this also has a non-surgical option, the Ponseti method. We have congenital heart defects in which you have really no treatment. Hydroencephaly, where you have a buildup of cerebrospinal fluid in the brain. This has a prevalence risk of anywhere from 0.5 to 2.5. So again, it depends on the risk factors being passed on. And then we have neural tube defects, which can have surgery. Some are going to be, of course, born dead or die within a few days. And folic acid replenishment can help that. Pyloric stenosis, as we've talked about before, can be treated with a surgery. And it has a prevalence of about 3 out of every 1,000 births. Now, apart from those, we are going to now focus more on adult disorders. So we have cardiovascular cancer and diabetes. And diabetes, of course, can be both adult and child disorders. So heart disease is going to present at about 25, or it's going to cause about 25% of all deaths of the United States, and coronary artery disease is the most common. And so this is just showing what's happening with coronary artery disease. You have your muscles in your normal artery, and you have your blood flow, and with an accumulation of LDL cholesterol, you get what's called a fatty streak. And fatty streaks are going to be macrophage foam cells that are eating or taking up the fats and cholesterol, not able to break them down fast enough, and so they form sort of this little micro tissue within the blood vessel, and that's going to promote inflammation. And so the inflammation will lead to a necrotic core. So necrosis means death. So you're having, with any inflammation, you have a neutrophil influence. So neutrophils come in and they basically are killing themselves, and they create this necrotic core. And then you're going to have fibers laid down to try to repair and also to wall off this necrotic tissue. And so you start to get a fibrous cap. And then you get a larger necrotic core as your cells die more. And then eventually you're going to get a thrombus, which is a ruptured plaque. And the thrombus, of course, can lodge itself into smaller blood vessels, and it causes a blockage. So risk factors for coronary artery disease are going to be obesity, smoking, an increase of cholesterol. If you have a first-degree relative that had coronary artery disease, and you have a higher risk when the first-degree relative is the female, because they have the higher threshold, similar to pyloric stenosis. And so it means you've inherited more risk factors if you have a first-degree relative that is female, or you have likely inherited them. So one of the things that is to point out is these blockages are a result of your macrophages, your immune cells, trying to remove fat, and they get overburdened because there's too much around. So it goes back to our story of you have your chylomicrons that we've been talking about from biochemistry. You have your chylomicrons that are going to deliver dietary fat. You have your liver that's going to package up the fat, your excess sugars into fat, and the VLDL particles. The VLDL particles are, of course, going to eventually be LDL particles. We've talked about LDL particles. They are going to be important in moving cholesterol around, especially to deliver it to cells that need it, such as those making hormones. However, if they accumulate, they're going to be modified and eaten by your macrophage. and then your macrophage becomes fat laden. And your macrophage is going to be engorged and become a fatty cell. Then eventually you'll get more macrophages forming a fatty streak that we just talked about. So you do have CoA cholesterol acyltransferase, and that's going to be important for esterifying your cholesterols and keeping them inside of your low-density lipoprotein particles. And so you can think about this whole process of moving around fats and any sort of disruption of this process, whether you have a defect in forming your very low-density lipoprotein particles, you'll get a fatty liver. If you have a defect in actually taking up the particles, the LDLs, the IDLs of your receptors, because you have receptors on the surface of your liver, for example, and those are going to be important for removing these particles. If you have any sort of defect in those receptors, that's also going to lead to a risk factor for having coronary artery disease. So with respect to coronary artery disease, there's been a focus on, of course, these proteins involved in lipoproteins. So we have apolipoproteins. We talked about them. We have different versions of them depending on the particle. And so these are some known defects in the apolipoproteins that have been associated with risk factors for coronary artery disease. You have lecithin cholesterol acyltransferase, and so that's going to be part of your high-density lipoprotein, important for moving cholesterol around. And so if that is defective, then your cells become cholesterol-laden because HDLs, remember, are the ones that go around and remove cholesterol from cells throughout your body. And if HDL doesn't do this properly, your cells eventually will shed cholesterol and get into the bloodstream, and then your macrophages will come along and eat them. And again, you can start to build up that fatty streak. So different apoproteins are going to have different influence. We have apolipoprotein C2, as we know, is going to activate lipoprotein lipase. And again, if you have a loss of function of this protein, then you're not going to have the release of fatty acids to be picked up by your muscle or by your adipose tissue, for example. Apolipoprotein E is a ligand for LDL receptor. And again, it goes back to these particles need to be retaken up by the liver so that they don't accumulate in the bloodstream. And if you have a defect of your apolipoprotein E, it cannot bind to the low-density lipoprotein receptor on your liver, and it cannot be, that particle will not be removed from the blood as efficiently. So there are many different lipoproteins that can potentially be affected. We also have our LDL receptor. So that can be defective. So it's not the apolipoprotein E that's defective, but in this case it would be the LDL receptor. We can have lipoprotein A, which is important for cholesterol. Lipoprotein lipase, the actual enzyme that's going to help to remove those fatty acids, can be affected. You have your lecithin cholesterol acyltransferase, which can have a defect, and so that means that you're not going to get esterification of your cholesterol. Your cholesterol ester transfer, which is going to be transferring cholesterol between lipoproteins. So this is where you have cholesterol esters accumulate in your high-density lipoprotein. They transfer it to the low-density lipoprotein that then takes it to the liver so the liver can deal with the excess cholesterol. And so if that's affected, then you can have a defect in moving around your fats. Others are going to be environmental factors. So you're going to increase your risk if you have obesity, because that makes sense because you have more fat. So if you have any of these risk factors, loss of function of any of those genes I talked about, and an increase of taking in fat and making fat, then you're going to increase your risk for coronary artery disease. Decreased risk are going to be exercising, healthy eating habits, and not smoking. Now with cardiomyopathy, you're going to have, this is where you have your heart muscles abnormal. And so over 100 genes have been linked to risk factors for cardiomyopathy. Um, and with hypertrophic cardiomyopathy, you have a thickening of the left ventricle. This you have beta myosin, which in the heavy chain is going to have about 35% of risk in familial cases. Myosin binding protein C is about 20%, and troponin T is about 15%. And so here we have, we're just going to go through a few of these. We have cardiac beta myosin heavy chain. And so this is important, of course, in thick filaments. You have actin binding ATP hydrolysis. And so if you have a loss of function, then you're going to have reduced force by your sarcomere. Your cardiac troponin T, and so cardiac troponin T is found only in the heart muscle. It's one of the three proteins that are going to make up troponin protein in your cardiac muscle. And so it's important in regulating muscle contraction in response to elevations of calcium. And so again, a loss of function of that would reduce force again. There's also myosin binding protein C. This is going to attach, this is going to be your basic unit associated with the sarcomere. It attaches the thick filament and prevents them from being broken down prematurely. So it's going to help to protect those thick filaments. And so if the thick filaments break down, if the myosin binding protein C is not able to protect them, you'll have more of the thick filament breaking down, which means you have to make more. And your sarcomere can become damaged. Your myosin binding protein C is also going to regulate how fast the muscles contract, and the mechanism for that particular function is not completely known. So you have dilated cardiomyopathy, which means you have an increase in size, and leads to the impaired contraction of ventricles. This is about a third familial. The most common is going to be autosomal dominant, and some X-linked or mitochondria influences as well. You have a Titin gene mutation, which is going to be the most common. We also can have desmin-affected actin, and of course your troponin can also be influencing this. So for dilated, we have our familial, which are listed here, the different genes that can be involved. It goes back again to those three proteins that were also previously implicated. You have your troponin T, cardiac troponin T. And so I'm not going to go through that function again, but just keep in mind that this is one of the proteins that that form the troponin protein complex in cardiac muscle, and this is to regulate contraction in relation to calcium. We have our titin, which is, of course, a major protein of muscle. We have our cardiac beta myosin heavy chain, and this is going to be making the beta myosin heavy chain important for actin binding, ATP hydrolysis. It's important in thick filaments, etc. We also have our beta sarcoglycan, and we have our delta sarcoglycan, as well as our dystrophin genes. And so you have your extracellular matrix, which is very important in giving strength to the muscle. And so these are proteins that are involved in interacting with the extracellular matrix. They're extracellular matrix proteins. So you can see here that they are all involved in stabilization, because mutations in them lead to destabilizing the contraction unit in your cardiac muscles. So sarcoglycan, the beta subunit, is important in anchoring your muscles, and it helps to maintain muscle structure. It stabilizes this trophin complex, which is important again in muscle strength. And then we have our delta complex, which is another subunit of the sarcogland. So again, it has to do with anchoring. And then, of course, dystrophin, which is the largest known gene protein. And it's in muscle cells, skeletal, and cardiac. It's part of a complex that strengthens muscle fibers, also protecting them from injury. And it connects the muscle fiber framework to the extracellular matrix. So sarcoglycans, beta and delta, are going to interact with dystrophin, and they're going to help to strengthen the muscle. And just keep in mind that the sarcoglycans are part of the extracellular matrix, and they are going to be closely related to the dystrophin protein. So any mutations in any of those three are going to lead to destabilization. Now, they're also thought to be involved in signal transduction. So that's why it says there decrease in signal transduction, because again, they're part of the extracellular matrix. Communication is important in coordinating muscle contractions. And so again, destabilizing these proteins can lead to a decrease in signal transduction as well. Okay, so QT interval. So one thing to keep in mind for QT interval is that this is the electrocardiogram, is that it is inversely proportional to the heart rate. So the QT interval will be short when the heart rate is fast, and the QT interval lengthens as the heart rate slows. So if you have an abnormally prolonged QT, this is associated with an increased risk factor with ventricle arrhythmias. If you have congenital short QT syndrome, this is associated with an increased risk of proximal atrial and ventricle fibrillation, which can lead to, unfortunately, sudden death. So you also have an autosomal dominant form, Romano-Ward syndrome, which is a loss of function of potassium channels. There are many genes associated with potassium channels, all shown here. The K indicates potassium. And so you have the CNQ1, CNH2, CNE1, etc. So you can have loss of function mutations of any of those. Or alternatively, you can have a gain of function of sodium or calcium channels. And so the sodium channel is the S, and the calcium channel has the CA. And so Romeo Ward is going to, with any of those, either K channels, sodium, or calcium channel defects, is going to cause disruption of the normal rhythm of the heart. So it's going to lead to arrhythmia. It also, the long QT syndrome of cardiac muscles, it means that you have to longer than would be necessary to recharge between your heartbeats. So again, here are some of the genes involved in long QT syndromes. You have your potassium channels, you have your sodium channels. Some of them are anchoring proteins, so you have your anchorin B protein. You have your beta subunit of the potassium channel, etc. And so they're all going to lead to prolonged QT intervals and arrhythmia. Okay, so moving on, we have hypercholesterolemia, which is an autosomal dominant familial disease. It's going to be implicated as a cause of about 5% of the myocardial infarctions, and this is in people less than 60 years of age. About 1 in every 500 are heterozygote for this. So what this leads to is 2 times the cholesterol levels of normal. You have exanthems, which are going to be fat cholesterol swellings that can present on the hands. Males with a familial hypercholesterolemia are going to have a higher incidence of developing coronary artery disease than they have about 75%. And then 50% of males are going to be fatal by 60 years of age. Females have a lower incidence of coronary artery disease, only 45%, and 15% fatal by 60. So again, this goes back to threshold. If your females have a higher threshold to present with disease, if they are the proband, then it increases the risk to other siblings. Now, if you have a homozygote, so these are heterozygotes, have 2 times. A homozygote will have 3 to 4 times the normal cholesterol levels. And so this homozygotes are, of course, going to have much higher levels of cholesterol that will have to be managed. So your low-density lipoproteins are very important, again, and they're going to be the overall remnants of your very low-density lipoprotein particles made by your liver. And they have a receptor. They typically go back to the liver to be removed from the circulation. So the low-density lipoprotein receptor is very important. And similar to what we talked about with cystic fibrosis, there are different types of mutations that are known with respect to this receptor. There are over 1,000 mutations identified. About 2/3 of these are going to be nonsense or missense, and the rest are going to be insertions or deletions. And it's thought that the insertions or deletions are caused by unequal crossing over of the ALU repeats. Remember, we talked about repetitive DNA, and those ALUs repeats were part of that repetitive DNA. Now, the different classes are going to be similar to what we talked about with cystic fibrosis, that you're going to have some that are going to have more deleterious effects. So for class 1, you absolutely have no protein produced. A heterozygote will produce half the number of normal receptors. A homozygote will produce none. A type 2 is where you make the LDL receptor, but it doesn't lead, so it gets degraded faster. So you make it, but you cannot get it onto the surface. Your LDL receptor gets to the surface, but it doesn't bind to the particles, so the ability to bind to LDL is lost. In 4, the receptor is normal, but it goes to the surface at areas that are not in clathrin-coated pits, and therefore, when the LDL binds to the receptor, it could not be taken up because it needs to be in those areas that can form vessels. And then 5 is where the LDL receptor binds and takes up the particles, but it can't dissociate, so it's going to get degraded. So familial hypercholesterolemia has different proteins that are involved. You have the ApoB proteins, and that can lead to elevated low-density lipoproteins. That receptor we just talked about, that can have different mutations, and in any defect of taking up the LDLs are going to lead to elevated LDLs. There is also protein convertase subtinsulin connexin type 9, and this has a role in actually degrading the LDL receptors. And so this will actually take up the LDL receptors in your liver and degrade them. So if you have a gain of function mutation, you're going to degrade your LDL receptors much faster, and so that's going to lead to an elevated LDL. If you have a loss of function mutation, it's going to degrade your LDL receptors slower, and so you're going to pick up more LDL, so you have reduced LDL in your circulation. So there are drugs that are targeting this particular protein to try to decrease the amount of LDL in the circulation, and they have been pretty successful at decreasing about up to 50% of your actual LDL circulating. All right, so now stroke is where you have a loss of blood flow to the brain, and an ischemic stroke is going to be obstruction of arteries, and that's about 80% of the causes of stroke. A hemorrhagic stroke is where you have breakage, and that's going to be less common. There are familial clustering of stroke, so if a parent has had a stroke, then there is an increased risk for offspring having stroke, about 2 to 3-fold. There are associated single gene disorders, so sickle cell anemia can increase your risk for stroke. You have some mitochondria genes, some mutations of Notch3 mutations that can all lead to an increase of stroke. Coagulation factors can be influence stroke, so we have protein C and protein S. These are inhibitors of the coagulation cascade, so if they have a loss of function, then you're going to have enhanced coagulation, which can lead to obstructions. You also have clotting factor 5 mutations that can lead to obstructions and stroke. Hypertension, which is just high blood pressure, is going to be a risk factor for heart disease as well as kidney disease. And so you have a hereditary systolic and diastolic blood pressure influence, and it's about 30 to 50%. Monozygotic twins are about 60%, so that means that there is a pretty substantial influence of genetics on hypertension. However, it's not 100%, so this also means that there are other factors that are influencing. And so it's thought these other factors are the amount of sodium, the amount of physical activity one does, the stresses one is exposed to, as well as, again, obesity. All right, so with respect to hypertension, you have your angiotensin, which is going to be released by your liver, and that's going to be converted by your renin to angiotensin 1. And then you have angiotensin converting enzyme, which is ACE, and we know we have ACE inhibitors. This is going to convert angiotensin 1 to angiotensin 2. So ACE inhibitors are blocking this transformation. Angiotensin 2 is going to be able to bind to your receptor, angiotensin receptor 1. There are also drugs that will be antagonists, so compete for the binding of the angiotensin 2. And the receptor, once it's stimulated, is going to release the hormone. And the hormone aldosterone is going to cause sodium retention and vasoconstriction, which increases blood pressure. And this is a feedback to kidneys to decrease renin secretion. And so the kidneys will decrease renin, and it will prevent this formation of angiotensin 1. So for hypertension, there are some single gene disorders that are very rare. There's Little syndrome, and so this is a sodium channel gene that is affected. We have Gordon syndrome, and this is going to be kinases that are going to lead to higher potassium, an increase of salt reabsorption. And there are thought to be somewhere up to about 20 genes linked to these rare single gene disorders. So again, these are rare, meaning that the more common is going to be the familial with the environmental risk. Okay, so now moving on into cancer. Cancer is, of course, becoming one of the most leading causes of death in the United States. With respect to breast, colon, prostate, and ovarian, there is familial clustering, but keep in mind most cancers are going to be spontaneous. And so you're going to have a lot of environment influences on cancer. So smoking, your diet, infectious diseases, you have your true cancer-causing viruses, the human papillomavirus, and then Hep B and Hep C are not true cancer causing. However, they cause such damage to your liver that your liver can become cancerous. We have proto-oncogenes and tumor suppressor genes. Proto-oncogenes are going to favor cell cycle progression. Tumor suppressors are going to prevent cell cycle progression. And keep in mind that proto-oncogenes are your normal genes. You need them. You need to have cell cycle progression. However, they're called proto-oncogenes because if they get a gain of function, then they become cancer, the potential to become cancer-causing. So types of cancer, we have sarcomas, carcinomas, lymphomas, etc., and I list them all here. Gliomas are glial cells of the central nervous system. Your sarcomas are connective tissues, carcinomas are epidermal tissues, and lymphomas and leukemias have to do with your lymphatic hematopoietic organs. There is a two-hit hypothesis for all cancers, and retinoblastoma is what was used to explain this. It's the Knudsen hypothesis. So if you inherited a hit, which means basically you have a good copy and a bad copy. So in this case, you have, I guess I can blow this up. So you have the pink, which is normal. The retinoblastoma mutation is this abnormal green. And so this individual inherited a normal and abnormal. So one green and one pink. Okay, so the mutation is already present in the germline when the baby was born. And so this mutation gets passed down through the genes. Now, the second mutation has to occur, and this is going to be a somatic mutation, not the germline mutation. And it's going to occur in some cells, and these cells are going to develop the tumor. Now, in another individual, they receive two good copies of the genes from the egg and sperm. And so the first mutation has to be somatic, that green one here. And then that second mutation is going to be somatic, and then you're going to get tumor development as well. So this is just to demonstrate that you have to have two mutations in order to develop your cancer. Now, this is a little bit simplified because you still have your tumor suppressor genes involved. And so it's thought that your proto-oncogenes need to be mutated, but your tumor suppressor genes also have to be mutated for cancer to develop. So tumor suppressors are just what they are. They're going to control cell division. So if your cell has a gain of function of one of the proto-oncogenes, your tumor suppressors are still there to make sure that you don't progress to the cell cycle unnecessarily. So if you get mutations in your tumor suppressor genes, then you can imagine a gain of function of a proto-oncogene would lead to uncontrolled growth and to tumors. So for your tumor suppressor genes, inheritance is going to be dominant in an individual, meaning that you're likely to get that second hit and you're likely to get some form of cancer. Inheritance of the mutation is, however, going to be recessive in a cell, meaning that each cell is going to be a heterozygote. It's going to express one good copy of that gene, and so each cell, in theory, is not going to lead to disease unless they get that second hit. So that retinoblastoma is a tumor suppressor gene. You also have APC, which is involved in Wnt signaling. Mutations in APC have been associated with polyposis disease or tumors. You have neurofibromatosis 1 and neurofibromatosis 2 genes, and these are going to be important in controlling RAS. RAS is, of course, a proto-oncogene because if RAS is on, it leads to growth. And so your neurofibromatosis will control that. And if you lose neurofibromatosis genes, then you're going to lose the ability to control RAS if RAS gets mutated. Neurofibromatosis 2 is going to regulate cytoskeleton proteins. All right, so proto-oncogenes are normal, not mutated genes. The proto-oncogene becomes an oncogene when you have a gain of function mutation. Sorry, that should be an N. And then when it becomes an oncogene, that means it's always on and you get uncontrolled growth. And this leads to what is referred to as a transformed cell. And this can be cancerous. So there are certain gene mutations that are important for breast cancer. We have BRCA1 and BRCA2, BRCA1, BRCA2, and these are going to be involved in DNA repair, so they're tumor suppressors. TP52, CHK2, and PTEN are also proto-oncogenes. They're tumor suppressors, and so loss of function of any of these can potentially lead to breast cancer. However, it's important to point out that 90% of breast cancer is not inherited. However, if you have inherited one of the known mutations, the likelihood of you getting that second mutation and an overall loss of function increases. So it's important to know if you have in your family history BRCA1 or BRCA2, for example, mutations. The environmental risk factors are never having children or having your first child after 30 years of age, alcohol use, high-fat diet, or estrogen replacement therapies are all going to increase your risk for breast cancer. Colorectal cancer is going to be about 1 in 20 Americans. It's second to lung cancer as far as prevalence in the United States. And again, most are not inherited. 90% are not inherited. You do have familial clustering, and your risk factor of, if you have a first-degree relative with colorectal cancer, you increase about 2 to 3-fold over the general population. The familial, again, can be caused by that APC gene, that tumor suppressor gene, and it's thought, or you can have a defect or mutation in the mismatch repair gene. So keep in mind that these are, the DNA mismatch repair gene is very important in repairing if there is a mismatch, and if you have a loss of function, that can lead to cancer, and that has been associated with colorectal cancer. You have an STK11, which is a kinase, which is autosomal dominant inheritance pattern, mutations in that are being passed down, and that's a Peutz-Jeghers syndrome. Juvenile intestinal polyposis is where you have 10 or more polyps before adulthood, and it's been associated with a serine threonine kinase gene. So remember, serine threonine kinase receptors are involved in growth. It also has other genes that have been involved in that. P10, which is a tumor suppressor gene, as well as SMAD4. Remember, SMAD is downstream of those serine threonine kinase receptors. Prostate cancer is going to be the second common cancer in men, and also second most common cause of death in men. Lung is, of course, first. So you have what's interesting about prostate cancer is you have a loss of heterozygosity, which is found in the tumor cells. And this is different than most cancers. Most cancers are heterozygote, meaning that they look very different. So this means that your tumor cells in prostate cancer are pretty homogeneous and look very similar. There is a polymorphism associated at chromosome 8 on the Q arm at position 24. This is a region of the chromosome that doesn't have any coding regions. However, mutations in this might influence down a gene that's close to it. It's a proto-oncogene MYC. And so if you have a mutation in here, in this region, that leads to turning on the MYC proto-oncogene, then you can potentially lead to cancer. Now, diabetes mellitus is type 1. This is an autoimmune disease. This is caused by the activation of autoreactive CD4 T cells. You also are going to generate autoantibodies against your beta cells of the pancreas. It's about a 10% clustering familially with respect to the insulin gene polymorphism. There is also a polymorphism with CTLA4. CTLA4 is an inhibitor of T cells, so a loss of function would lead to can lead to an increase of autoimmune disease. There's also a PTPN22 gene that is again involved in T cell regulation, and so again a loss of function of that would lose control of your CD4 T cells. Your type 1 autoimmune disease is thought to be associated with the human leukocyte antigen. So 90% of whites have what's called an HLA-DR3 or HLA-DR4. We also have an HLA-DQ, and at position 57, absence of aspartic acid makes you more susceptible. So homozygotes with this HLA-DQ phenotype with that loss of the aspartic acid have 100% times likely to get diabetic. And so what this is telling you is that the human leukocyte antigen is important for activating your CD4 T cells. So you have your macrophage, and it has on its surface the human leukocyte antigen. So here's the HLA. And within this human leukocyte antigen, you can have your antigen. And the antigens can be a flu antigen, it can be a cold antigen, it can be a SARS antigen. But also these HLAs present normal proteins that are in your body. So in this case, this would be a particular antigen from the beta cell of the pancreas. And so your HLA DQ, or your individual with HLA DQ with that susceptibility is going to present this, and it's going to activate your CD4 T cell. A lot of people who don't have this phenotype would not present this particular antigen and therefore would not activate their CD4 T cell. And we'll learn more about how this happens. HLA molecules anchor antigens based on differences within their grooves, within their pockets. And so by losing the aspartic acid, you're allowing this particular HLA to bind to the beta cell protein. And most people that have that aspartic acid, you would not allow the HLA to bind to that beta cell protein. So it would never present it to the CD4 T cell, and the CD4 T cell would never get activated. Now, type 2 diabetes is referred to as a metabolic disease. And so this is going to be more prevalent, and it's more prevalent now with our lifestyle. With type 2 diabetics, it has no HLA association. So what we just talked about, that HLA has no HLA association, no antibodies, no T cells, no autoimmunity. In this case, unlike type 1 that cannot produce insulin, type 2 can produce insulin. So there is insulin being produced, and it can be treated with diet modification as well as drugs to help to remove the sugars from the circulation. This is referred to as insulin resistance, and you can often think of it as a car. If you're constantly turning on and off, or your engine in an older car, you're going to wear out your ignition. And so that's the same thing with insulin receptors. If you're constantly having insulin present, downregulate receptor, upregulate receptor, downregulate constantly over and over again, you kind of just wear the system out. So you tend to become more insulin resistant. The risk factors are going to have an increase with a positive family history of type 2 diabetes, as well as environmental risk factors such as obesity. There are about 70 genes that have been linked to an increase in risk factor. So you have your ability to secrete insulin. You have your peroxisome proliferator gene. This is important for your receptors. You have potassium channels, which is very important for insulin secretion again. So mutations in the potassium channels can also lead to an increase of risk. Now there's also maturity onset diabetes of the young. This is not associated with obesity. It occurs before 25 years of age, and it's thought to be an autosomal dominant inheritance pattern. 50% of the people who suffer from this have mutations in glucokinase. And you know that glucokinase is the variant of hexokinase that's in the liver as well as in the beta cells of the pancreas. So a loss of function of glucokinase would impact your beta cells of the pancreas, because when there's a lot of sugar around, your glucokinase would not be increasing ATP enough to get that potassium channel shut so you can get the calcium channel open and have release of insulin. So it would impact the release of insulin. So that the actual insulin, the ability to release insulin is there. It's just that the glucokinase is mutated. There are other mutations that can influence this as well. You can have your neurogenic differentiation factor 1 again be influenced. Insulin promoter factors can be affected, so your beta cells can't make enough insulin, for example, from as a protein. So there are other mutations as well.